Pleasure to introduce Dr. Paul Cohen, who's visiting us today from Rockefeller University. I thought I had this brilliant and original idea to invite Dr. Cohen after I perused the Endocrine Society program for this upcoming Endocrine Society, and I saw that he was giving one of the big sessions on a very interesting topic, and he sounded like an intriguing speaker, only to find out that he's well known to the metabolism group here and gave a talk here last year to the metabolism group. So there goes my originality. But he was gracious enough, I called him only about 2 months ago, to say, yeah, that works for me, March 1st, that's good. And we always appreciate that in speakers, especially speakers as accomplished as Dr. Cohen. So he has an MD PhD. He got his PhD at Rockefeller University, his MD at Cornell, and then went on to do residency, I believe, at Columbia in internal medicine. He did a cardiology fellowship up in Boston at the Brigham. And then that was followed by a postdoctoral fellowship at the Dana-Farber, I believe. He's been blue blood trained, not just in terms of his universities, but in terms of his mentors. So one of his mentors was Dr. Jeff Friedman at Rockefeller, who we know him as the other Friedman brother, because Scott Friedman, his, I believe, younger brother, is the head of liver here. And then with Dr. Spiegelman, I believe, up in Boston. He has been working on, since his PhD, on the enigma in terms of there are some fats that are really bad for us and there are some fats that are really good for us. And in that regard, he has really accomplished a lot. He has many, many different grants and many different awards for somebody, you know, fairly junior in his years and in his career. And in perusing his resume, bear with me here, I was reminded of the Steve Martin movie, The Jerk. Okay, but stick with me on this one. You know what I'm referring to? He's leaving. No. Because when I was looking over the work that he does, one of my favorite scenes in that movie is when Steve Martin goes to a restaurant. He's trying to impress his girlfriend, which is Bernadette Peters, and he's never been in a nice restaurant, and they come over and they offer him some wine. He said, well, I'd like to see your wine list. I'd like to order some wine. And they said, really, red or white? And he goes, oh, it comes in colors? He said, then I would like your best beige wine. So it turns out, I learned this morning, that fat comes in white, brown, and beige. Okay, so we're really looking forward to this talk, and I apologize for the reference.
Well, thank you for the kind invitation, and it's great to be here. I am a big Steve Martin fan, but that reference had eluded me. So now I've learned something new. I actually also wanted to point out, as a medical student after I finished my PhD, I did a rotation in endocrinology at Mount Sinai, and I actually interacted with Dr. Gabrilov, so it's really nice to be back here. And although I didn't go into endocrinology, many of the things that have shaped my career have involved areas of prevention that are not really that dissimilar from what many of you do clinically or research-wise. So today I'm going to talk about transcriptional control of adipose tissue phenotype, and I've tried to provide a different talk to the one I gave when I visited a year ago, so if any of you were here then, hopefully this will not be too boring for you. So as many of you know, obesity is associated with a constellation of health problems, and I've highlighted several in red that are prominent features of at least my clinical practice and probably those of you here as well. And just to give you a fuller sense of the scope of the problem, in the United States now, 1 in 3 adults are obese and 2 in 3 are either overweight or obese, and this also affects nearly 20% of children. This costs our healthcare system more than $100 billion each year, which is clearly unsustainable. One area that's become of interest to me in my lab recently is the link between obesity and cancer. And obesity is now overtaking tobacco as the leading preventable cause of cancer. And overweight and obesity have been estimated to account for as many as 20% of cancer deaths. And lastly, this is now no longer a problem of the United States or of industrialized nations, and more than 2 billion adults worldwide are either overweight or obese. And for the first time, illness due to overnutrition has actually eclipsed illness due to undernutrition. And so my interest for a long time since my PhD years, both clinically and research-wise, is in understanding how obesity impacts health. And why is it that some, but not all individuals with obesity are afflicted with conditions like diabetes, fatty liver disease, hypertension, heart disease, and cancer, whereas others are unaffected? And so in my presentation today, I'll cover several areas. I'll first talk about the links between fat phenotype and metabolic health. I'll then talk about the transcriptional regulation of adipose phenotype. I'll then talk about some new data from my group looking at the role of fat phenotype in obesity-associated diseases, and this will focus on hypertension. And then lastly, I'll talk to you about a new approach we've been taking to try and more fully dissect the endocrine functions of adipose tissue. So to start, I thought I would illustrate this problem with a clinical vignette, and these are two patients who I saw when I was still in Boston and focusing on preventive cardiology. So these patients represent two extremes of spectrum that I'm sure many of you have seen. So both women, almost the same age, almost the identical weight, both with BMIs above 50, but very, very different phenotypes. So patient number one came in to see me. She had uncontrolled hypertension, uncontrolled diabetes, hyperlipidemia. When I met her, she was on 15 different medications as well as insulin, and went on to require multiple hospitalizations for congestive heart failure and then renal failure requiring dialysis. And I don't think it's a stretch to say that her life expectancy is probably could be measured in a number of years. This patient actually weighed a few pounds more, came in to see me for a preoperative evaluation before bariatric surgery, and was quite healthy. She had normal blood pressure, normal lipids, only borderline diabetes, and really the only thing that bothered her was her knees. And she went on to have a sleeve gastrectomy with no issues, lost nearly 100 pounds, and continues to do well. So what's the difference between these two individuals? And of course, this is a complex question, and there is not one single factor that explains it, and there are undoubtedly contributions from things like diet, nutrition, and genetics. But one really important determinant is where these people store their excess fat. And many, many, many epidemiologic studies, as well as clinical reports dating back to the 1940s, have shown that individuals who store their excess fat in a visceral location, or this so-called apple-shaped obesity, are more likely to be metabolically unhealthy than those who store their fat in a subcutaneous or pear-shaped distribution. And data has shown that people who store their fat in a visceral location, like patient one, are more likely to be insulin resistant and diabetic, to have hypertension, dyslipidemia, cardiovascular disease, cancer, and even mortality. And I'm not going through all of these epidemiologic studies, but I'm happy to discuss them if anyone has any questions. And the other important point I want to make is that although in the initial description these were linked with sex hormones, and this was referred to as android obesity and this is gynoid obesity, and there are undoubtedly roles of sex hormones in how body fat is distributed, there are substantial numbers of people of both genders who store their fat in either distribution. So in my research, I've been interested in understanding the cellular and molecular mechanisms for this phenomenon. And to do that, we've taken an increasingly reductionist approach to move from how fat is distributed to the cellular and molecular phenotype. So on first pass, if you look at the tissue and the cells, you see fat cells. And so one argument could be that fat is fat, and there's no difference. But work done by many people in the field, including Susan Fried, who's here, has shown that that's not actually the case, and there are distinct differences between fat cells, many of which persist ex vivo. So rather than fat having its phenotype based on where it is in the body, there are clearly cell-intrinsic differences in fat in different locations. And one thing that we've come to understand over time is that fat in a visceral location is more likely to develop this chronic inflammatory state, whereas fat in a subcutaneous distribution not only is less likely to be inflamed, but is able to dissipate energy in some cases because it contains these thermogenic or energy-dissipating fat cells that I'll talk more about in a minute. So that leads me into the cellular phenotype. And one of the main players of adipose tissue, of course, is the fat cell or the adipocyte. And most of us, when we think about fat, we think about white fat cells. And these are cells with a very large lipid droplet and few mitochondria. And these cells have evolved to efficiently store energy in the form of triacylglycerol. And this energy can also be mobilized by lipolysis when nutrients are needed. But mammals also possess brown fat, which has a very different phenotype. So brown fat has these numerous small lipid droplets that allow the lipid to be more readily mobilized. They have a very, very high number of mitochondria. They're among the most mitochondria-dense tissues in the body. In fact, the iron in mitochondria actually gives the tissue its brown color, so the fat is literally brown. And because of these properties, these cells are able to actually dissipate energy, so they can convert chemical energy into heat. And the way they do that is depicted here. So this is a depiction of the mitochondria showing oxidative metabolism, which might give you flashbacks to your biochemistry days. But what I'm showing here is the standard pathway of oxidative phosphorylation, which results in the production of ATP. Reducing equivalents are passed along the electron transport chain, and this generates a proton gradient, which is normally used to power the synthesis of ATP. What is unique about thermogenic fat cells is that they possess this protein called UCP1, or uncoupling protein 1, which sits in the inner mitochondrial membrane and allows the proton gradient to leak back across the inner membrane. As a result of that, this gradient is dissipated, and so rather than being used to power the synthesis of ATP, this chemical energy is dissipated in the form of heat. And so brown fat is thought to be critically important to maintaining body temperature, and if you think about it, especially in small mammals and newborns, when body surface area is relatively high relative to body mass, and humans and small mammals are born hairless, maintaining body temperature is absolutely critical to early survival. So now let's move a little bit more reductionist to the molecular components that are involved. So both white and brown adipocytes require this transcriptional protein called PPAR-gamma, which is a nuclear hormone receptor. Some of you may be more familiar with it because it's the target of the thiazolidinedione drugs that used to be widely clinically used. Of course, now they're very seldomly used because of different side effects. But PPAR-gamma is expressed in all fat cells and is both necessary and sufficient to make a white or a brown fat cell a fat cell. And this is work on the left done by my postdoctoral mentor and Peter Tontonos when he was a student in his lab. And what they showed was that simply putting PPAR-gamma into a fibroblast was sufficient to endow that cell with the properties of a fat cell. This is oil red O staining, showing that PPAR-gamma can allow these cells to differentiate and take up lipid, and this cannot be achieved with other nuclear hormone receptors like PPAR-alpha or RXR-alpha. And then in loss of function studies, this is an in vitro study showing that if you delete PPAR-gamma in these adipogenic fibroblasts, you lose the ability to differentiate into a fat cell, so no longer have any oil red O staining. And then a number of labs, including Bruce's lab and Ron Evans' lab and others, generated mice with germline deletions of PPAR-gamma, and these mice lack both white and brown fat. So shown here is the interscapular brown fat, which is the main depot of brown fat in a mouse, and the knockout animal completely lacks brown fat. So this then leads to an obvious question. If PPAR-gamma is necessary and sufficient to make a fat cell, but I've just told you that white fat cells and brown fat cells almost have diametrically opposed phenotypes, what accounts for the difference? So clearly there must be other factors, and one really important factor is this protein called PRDM16, which has been a focus of my work. So PRDM16 is a transcriptional co-regulatory protein, meaning it doesn't itself need to bind DNA to exert its functions, but it binds to other transcription factors and coordinates the overall complex that governs what genes are expressed. And so in the work I'm showing you here, done by Patrick Seal, he identified PRDM16 as being highly enriched in brown fat relative to visceral white fat. And in fact, if you ectopically express PRDM16 in a white fat cell, it now gives that cell almost all of the properties of a brown fat cell. So shown here are a number of genes that are characteristic of brown fat cells. For the purpose of this talk, the names don't matter, but what you can see, I hope, is that expressing PRDM16 now induces all of these brown fat specific genes. And moreover, when he made a transgenic mouse that now expresses PRDM16 in all fat cells, you get the emergence of these small multilocular fat cells within white fat, and this is immunohistochemistry for UCP1, this key protein of brown fat, and now you have UCP1 staining within the white fat, which does not normally occur. There also is a third type of adipocyte, and this is something that we've increasingly focused on and was introduced in the Steve Martin movie. In addition to white fat and brown fat, mammals also have beige adipocytes. And these cells can also dissipate energy much like a brown adipocyte. The main distinction is that their activity is highly inducible. So whereas a brown fat cell can be thought of as being more constitutively active, beige fat cells have inducible activity. Without a stimulus, they may have a phenotype much like a white adipocyte, but in response to stimuli, with the most prominent ones being cold exposure, beta-adrenergic agonists, exercise, or thiazolidinediones, they can highly induce this thermogenic program. And we now know that humans have beige adipocytes, and because of their inducibility, they've become a very attractive therapeutic and pharmacologic target. So when I started my postdoctoral work, I was really interested in understanding the transcriptional basis for the difference between subcutaneous and visceral fat. Prior work had shown that, as I showed you, that PPAR gamma is necessary for all fat cells, and PRDM16 seems to distinguish white fat from brown fat. But I thought, well, what about subcutaneous fat and visceral fat? And so to address that, I did this screen, and this is just the two most promising candidates that came out of this screen. And what I did was I looked at all of the transcriptional components in the genome, about 2,000 different genes, and using a PCR-based approach looked to see which of those components are enriched in either subcutaneous fat depots or visceral fat depots. And what we found was that PRDM16, the same protein that I told you is brown fat enriched, is also expressed across a variety of subcutaneous fat depots, but expressed at only low levels in visceral fat. Interestingly, it's also highly expressed in fat around the aorta, and I'm not going to talk about that today, but that's something we're investigating further. We also became interested in this transcription factor WT1 that's most famous for being mutated in Wilms tumor. There was not previously known to have a role in fat, but WT1, as you can see here, is highly expressed across a variety of visceral fat depots and really undetectable in brown fat or subcutaneous fat. And so to go on and study the role of PRDM16 in subcutaneous fat, we used a genetic approach where we used the pre-lock system to make mice with a deletion of PRDM16 in fat cells. And we used the adiponectin pre-driver because it's mainly expressed postnatally. And something I haven't told you here is that brown fat, the sort of preformed brown fat in the interscapular location, forms relatively early during embryogenesis, whereas subcutaneous fat only begins to form late during embryonic development and continues to develop postnatally. And adiponectin is also expressed only late during development. And so our hope was that although we would delete PRDM16 in all fat cells, because the brown fat would have already formed, perhaps we wouldn't affect its function, whereas the actively forming subcutaneous fat would be affected by deleting PRDM16. So this is just showing you here at the RNA and protein levels that we were successful in deleting PRDM16 from fat. And then what we found was that by deleting PRDM16, we were able to completely ablate the function of these thermogenic beige adipocytes without affecting brown fat. So shown here is immunohistochemistry staining for UCP1, again, this key protein that marks thermogenic fat cells. And what you can appreciate is that in brown fat, the entire section stains for UCP1. When you expose animals to Cohen, you see more intense staining, and this is equally true in both control and knockout animals. But in the subcutaneous fat, you see something rather different. So in the control state, none of the cells stain, but in response to cold exposure, you see the emergence of these UCP1 positive beige fat cells, the inducible cells that I was telling you about. And this is completely absent in the knockout animals. No matter how many sections we look at, we just can't find any UCP1 positive cells in the absence of PRDM16. So we then went on to look at what are the physiological consequences of ablating these cells. And so we looked in both tissues, cultured cells, and the whole animal, and in each of these assays we're measuring oxygen consumption. And the main take-home point is that although oxygen consumption in brown fat is not affected, both basal and stimulated oxygen consumption. The subcutaneous fat is reduced when you delete PRDM16. This is true in tissue and in cells. And in fact, if you put the whole animal in an indirect calorimetry setup and measure whole body oxygen consumption, you get about a 10 to 15% reduction in whole animal energy expenditure when you delete PRDM16 and ablate beige fat. This results in effects on body weight. So on a standard chow diet, there's no difference over time. On a high-fat diet, you do see that over a long period of time, the knockout animals do gain more weight, but it's a relatively modest increase in body weight of about 5 to 10%. More impressive than this increase in body weight, though, is where the fat is coming from. So when you do body composition, all of the excess weight is accounted for by fat mass. And when you actually look at the individual fat depots, what you interestingly see is that when you knock out PRDM16, the inguinal depot, which is the largest subcutaneous fat depot in a mouse, is now about twofold greater in mass than in control animals, whereas the size of the other depots is unchanged. So we also went on to look at the fat more closely, and what I'm showing you here is just a section stained with H&E from control and knockout animal, and I don't know how well this reproduces, but I hope you can appreciate that in the control animal you see cells with a range of sizes, some small cells, some large cells, whereas in the knockout animal almost all of the cells are uniformly large, and this is shown quantitatively here. And we also dissociated the tissue and did FACS analysis to look at immune cell accumulation within adipose tissue. It's been a really growing area of research, now referred to as immunometabolism, showing that a whole host of immune cells accumulate in fat tissue and have important physiological functions. I also pointed out to you that visceral fat, at least in the setting of obesity, becomes inflamed. And one of the hallmarks of that is an accumulation of macrophages. So this is shown you here, macrophage quantitation in control and knockout animals, and there's about a threefold increase in macrophages in the knockout fat. We also looked at insulin and glucose homeostasis in collaboration with Jerry Schulman, and to do that we did hyperinsulinemic euglycemic clamp studies in animals after 6 weeks of a high-fat diet, so this was well before any difference in body weight or body composition. And what we see already is at baseline, the knockout animals have an increase in fasting insulin. And then looking at the whole animal by glucose infusion rate and glucose uptake, there's evidence of whole body insulin resistance in the knockout animals. And at the end of the experiment, we injected the animals with radio-labeled glucose, and we saw a significant reduction in glucose uptake in both subcutaneous and visceral white fat, no difference in skeletal muscle, and actually a slight but not statistically significant increase in brown fat, perhaps as an attempt to compensate for the defect in beige fat. And when we looked a little bit more closely, we saw that there was actually a somewhat interesting form of insulin resistance in these animals. So in many models of insulin resistance, at least in mice, and you all could tell me better in humans, muscle is really the tissue that accounts for the great bulk of glucose uptake. We saw no difference in glucose uptake in the muscle, but what we saw is that hepatic glucose production was significantly greater in the knockout animals than in the control animals. So in a normal state, when you clamp an animal and make it hyperinsulinemic, hepatic glucose production, as shown here, is dramatically suppressed. And this is severely blunted in the knockout animals, and with that, the animals also have hepatic steatosis with increased liver triglycerides. And so something that has occupied us then and continues to occupy us is, so what's responsible for all of these phenotypes? Is this simply just due to defective thermogenesis or this reduction in energy expenditure? Well, that's certainly possible, although more recent work from a colleague has shown that this probably does not explain the phenotype. So we have considered other possibilities, and I'll talk about two of them here, that are not mutually exclusive. So one is the endocrine function of these cells. There are many phenotypes, with the liver being one example, where simply ablating beige fat leads to effects in the whole animal and in other cell types and organ systems. And so one compelling possibility is that's due to endocrine mediators. And the other possibility, which I'll show you some data for, is that if you delete PRDM16, you may fundamentally be altering the phenotype of fat. And that's depicted here. So we refer to the phenotype at a molecular level as visceralization of the subcutaneous fat. So what I mean by that is we had already seen when we deleted PRDM16 that we now get larger fat cells increased macrophage accumulation, insulin resistance, hepatic steatosis. These are all features that you ordinarily just see in the setting of visceral obesity. But we're now seeing it in these animals, which actually have an increase of subcutaneous fat mass. But when we looked at the molecular markers of the fat, above this black line are genes that are normally only expressed in visceral fat cells, and the heat map in red and orange shows genes that are overexpressed. About a third of genes that are normally visceral fat selective are now highly expressed in the subcutaneous fat of PRDM16 knockout animals. And so what this is telling us is that by deleting PRDM16, we are now shifting the phenotype of fat towards a more visceral phenotype. And in fact, one of the genes that's most highly expressed is WT1, this visceral fat selective transcription factor. And so we think PRDM16 is a real important determinant of depot phenotype. So what we've gone on to do, and I'll sort of change gears now, is to use these mouse models to try and understand mechanisms linking obesity to associated diseases. And so we mainly use this PRDM16 knockout mouse that I've been telling you about that lacks beige fat as a model of visceral or unhealthy obesity. And then we also have a reciprocal mouse model, a transgenic mouse model that overexpresses PRDM16 in fat cells. These animals have enhanced beige fat function, and we use these as our models of healthy obesity. And so just to summarize what I've shown you with these two models, we know that the knockout animals have an ablation of beige fat. They have more unhealthy visceral type fat with increased inflammation, only mild obesity, but fairly prominent insulin resistance and hepatic steatosis. And then the transgenic animal in all regards seems to have the opposite phenotype with a mild protection from obesity, but increased insulin sensitivity. So what about other comorbidities associated with obesity? And one that we've increasingly focused on since I started my own lab is hypertension. And obesity, as many of you know, is a major risk factor for essential hypertension, and it's thought that obesity might account for 75% of the risk for hypertension, and risk is particularly elevated in individuals with visceral obesity. These are just some lines of evidence from prior work that I think establishes a rationale for looking at adipose tissue. So firstly, hypertension can improve with weight loss, and really only modest weight loss. So you can take an individual who's very obese, and if they lose 5 to 10% of their body weight, their blood pressure often comes down and is much easier to control. In addition, mutations in PPAR-gamma, this key transcriptional regulator of fat, in humans, mutations in PPAR-gamma actually cause a variety of phenotypes, including significant hypertension. Thiazolidine diones, which are pharmacologic agonists of PPAR-gamma, can lower blood pressure. And then lastly, lipoatrophic mice, which lack all adipose tissue, develop hypertension. So I think there's some rationale for looking at the links. And we were interested, well, what about in our mouse models? We have mouse models of healthy and unhealthy obesity. Is blood pressure altered in these animals? And so to investigate that, we've been using this system here. This is a radiotelemetric system where we implant a catheter in the mouse's carotid artery. This is connected to a radio transmitter that sits in the animal's body cavity. And what this allows you to do once the animals recover from the implant is you can record blood pressure in awake, freely moving animals, and the battery lasts about 12 weeks. So you can also collect longitudinal data over time and in response to different manipulations. This is preliminary and unpublished data, but what we are seeing in these studies is that the knockout mice which lack beige fat seem to have elevated mean arterial pressure, and our transgenic mice that have increased beige fat have reduced mean arterial pressure. Before the dashed line are samples done on the standard chow diet, and then after the dashed line is sampling on a high-fat diet. And I think the most prominent finding that we seem to be observing here is that the animals have alterations in blood pressure even without stimulation or challenging with the high-fat diet. So this, I think, is a nice entry point into this question of how fat communicates with other tissues. Are some of the profound benefits associated with the activation of thermogenic brown and beige fat mediated by endocrine factors? And secondly, are the deleterious phenotypes associated with visceral fat mediated by crosstalk with other organs? And there are a variety of endocrine factors produced by fat cells that are very, very well known, things like leptin, adiponectin, resistin, and so on. But what really caught our attention is that if you look at gene expression data from fat cells and run all of the genes that are expressed by fat cells through an algorithm that predicts what genes are likely to have a signal peptide and therefore have the potential to be secreted, what you see is that more than 1,000 genes are expressed by fat cells and at least computationally have the potential to be secreted. And if you then look at what those genes are, you see that about 20 of them have been very, very, very well studied, thousands of publications, but literally hundreds of genes have not been characterized in fat. So we think this represents a great opportunity for discovery, but we also feel that the computational approach has several inherent limitations. And so what we'd really like to do is not make predictions based on gene expression data, but actually measure levels of proteins in conditioned media from cells and in the blood of animals. And so to do that, we've been using a mass spec-based approach to actually categorize the proteome. And this has some inherent limitations, at least in the traditional approaches. So the way people would traditionally do this, if you wanted to characterize the proteome in either cells or in animals, would be to collect conditioned media or collect plasma or serum and do mass spectrometry and see what proteins are identified. One problem, though, is when you do that, you get an overwhelming signal from very abundant serum proteins like albumin, and this tends to drown out the signal from less abundant proteins that we're actually interested in that are in the nanogram or picogram per mil range. Now, if you're looking at cultured cells, you can say, well, why not just collect the media in serum-free conditions and see what's pumped out by the cells? And certainly you can do that, and that's been productive in many cases, but it's also become very clear that switching fat cells from media with serum, which they like and need to survive, to serum-free conditions profoundly alters the phenotype of the cells. So what we'd really like to do is characterize the proteome without taking away the serum. And so to do that, we've been using this approach called BONCAT that an MD PhD student in my lab has been using, and I'll take you through that here. So this is chemistry developed by a variety of investigators, mainly David Tyrell and Aaron Schumann at Caltech. And the idea is shown here. So in standard protein synthesis, methionine, for example, is recognized by the methionine tRNA synthetase and incorporated into a synthesizing protein. So there are chemical analogs of amino acids that can be generated, and one of them is shown here called azidohomoalanine. This is an analog of methionine that can also be recognized and incorporated by the methionine tRNA synthetase. But the really key distinction is that this analog contains an azide group, and azide groups do not exist in nature. And so when you add azidohomoalanine, what you get is an azide-labeled protein. The reason why that's useful is because you can then take advantage of this approach called click chemistry. So if you take an azide-labeled group and then expose it to an alkyne group in the setting of copper catalysis, you get this very, very robust covalent reaction. And you can use that reaction to cross-link newly synthesized proteins with an azide group and then pull them out of solution to do mass spec. And so the approach we've started out doing initially in cells, but now in animals, is you can culture primary fat cells and then at your time point of interest, change the medium, so you keep the serum in, but you remove methionine and instead replace that with azidohomoalanine. Collect the conditioned medium, which has these azide groups. You then collect the media and add an alkyne group that's conjugated to a bead. You get this covalent reaction. You then use the bead to pull out all of the newly synthesized proteins, digest them, and do mass spec. And so this is just showing you here some of our initial quality control data. So this is a Kumasi gel just showing protein loading. So you can see there's abundant protein in the media across all conditions. But now what we've done is rather than linking the azide group to a bead, we've linked it to a fluorescent marker. And you can see that we detect proteins only with azidohomoalanine. And if you actually look a little bit more closely, there are a number of proteins that are present in specific fat cell types. We're looking at visceral, subcutaneous, and brown fat cells in culture that are present in one cell type but not in another. And so we've now done several experiments, and I'll just take you through some of the data to give you a sense of what we're finding. So firstly, we decided to look at fat cells from either C57 black animals. This is a genetically inbred strain that is prone to obesity and insulin resistance, and AJ mice, which are resistant to obesity and insulin resistance. And we thought perhaps there are endocrine factors that might explain the differences in the phenotype between these animals. We identified about 800 proteins using this approach in the conditioned media. And what you can see here is that the great majority of the proteins fell along the diagonal, meaning they're at equivalent levels in both types of cells. But a number of proteins fall significantly off of the diagonal, including this protein, angiotensinogen, which I'll come back to soon, as well as a number of other novel proteins that haven't been characterized in fat before. So something we've recently completed is we've mapped the secretome of the three major adipose cell types. So we've cultured either visceral fat cells, subcutaneous fat cells, or brown fat cells, collected conditioned media, used this click chemistry-based approach, and now we are up to about 800 unique proteins. And when we use a heat map, we see that there's a pretty interesting pattern. So firstly, there are a number of proteins not shown in this heat map which are expressed in all three cell types, and these would include things like adiponectin, adipsin, and so on. But there are also proteins that are highly cell type enriched. So these would be, for example, about 40 or 50 proteins that are highly enriched in visceral adipocytes, subcutaneous adipocytes, brown and subcutaneous adipocytes, or just brown adipocytes. And we're now beginning to study the function of some of these proteins to see what they actually do physiologically. We've also started to extend this in vivo. So it's great to study what a cell can pump out, but that's not at all physiologic. You can also treat animals with these analogs, and they appear to be non-toxic. You can either give them by injection or by supplementing the diet or the drinking water. And so in this experiment here, we injected mice with either vehicle or azidohomoalanine, and we looked at either control or these PRDM16 transgenic mice, which have enhanced beige fat function. And we identified roughly 800 proteins, and we found a number of proteins in the blood now that are enriched between these different strains. So what we'd really like to do, and this is where the work is moving going forward, and we've just started to employ these mouse models, is to not just identify proteins that are in the blood. So one limitation of this approach is you can label proteins in the blood, but this doesn't tell you anything about where the proteins came from. You just, in principle, the azidohomoalanine can be taken up by any cell in the body, and any protein that's made and pumped into the circulation can be detected with this approach. What we'd really like to do is be able to collect plasma or serum from an animal in a particular condition and say, for example, what are all of the proteins in the blood that have been made by a fat cell or by a thermogenic fat cell or a muscle cell? So how can we do that? So that approach is actually now possible. We're just beginning to do it, so I'll take you through the approach we're taking. So again, using click chemistry, now there's a different analog to azidohomoalanine called azidonorleucine. I don't expect you to remember the structure, but one of the key distinctions is that this has a much longer and bulkier side chain than azidohomoalanine. And the consequence of that is that it cannot be recognized by the native methynyl tRNA synthetase, and so cannot be incorporated into the proteome. However, in this paper from Tyrell and Schumann, what they did was they began making point mutations in methynyl tRNA synthetase. And somewhat surprisingly, they found that mutating a single amino acid in the methynyl tRNA synthetase now allowed it to very efficiently take up azidonorleucine and incorporate it into the nascent proteome. And the reason why this is so valuable is because they have now gone on to make a mouse, which they've shared with us, that conditionally expresses this mutant methynyl tRNA synthetase. So in the basal state, there is a stop codon flanked by LOXP elements in front of the mutant methynyl tRNA synthetase. And what that allows you to do is to cross that floxed mouse to your cree line of interest such that you only express the mutant methynyl tRNA synthetase in that cell type. And so we're now crossing these mice to either adiponectin cree mice to label all fat cells or UCP1 cree mice to label thermogenic fat cells. And then the other advantage of this approach is that you only start labeling when you introduce the modified amino acid either into the diet or by injection. So you can also couple this to physiologic manipulations. You can, for example, look at what labels in either lean or obese mice, fed or fasted mice, sedentary versus exercise mice. You can think of almost an infinite number of combinations. So I want to come back to how we're going to deal with all of this information. So already just from our cell line work, we've identified about 800 proteins, and it's clearly not feasible in a single lab for us to work out the function of all of these proteins. And so how might we begin to think about which of these are most physiologically important and what their function is. And so one approach we've been taking is by using orthogonal data sets. So we've been cross-referencing our adipose-specific factors with RNA sequencing data and then doing different types of validation. And we've used that, at least initially, to think about mechanisms underlying the alteration in blood pressure in these models. So I told you that when you delete PRDM16, you have an increase in blood pressure, and when you overexpress PRDM16, can I have some, is there any water here? When you overexpress PRDM16, you have lower blood pressure. And so we wanted to understand what the mechanisms for that were. And what we saw here was that one of the genes that's most highly expressed is angiotensinogen. And we also saw that, thank you. Thanks. We also saw in our cell type specific proteomic data that angiotensinogen is highly visceral enriched. And then we also saw that angiotensinogen is highly enriched in the obesity-prone black 6 mouse relative to the AJ mouse. So we're now focusing more explicitly on whether angiotensinogen might explain the difference in blood pressure in our different mouse models. And so you all know this pathway, the Renin-Angiotensin-Aldosterone system, which is important in vasoconstriction and sodium uptake. And so we see increased angiotensinogen expression in our PRDM16 mutant mice. And
that's plasma or adipose tissue.
So we see increased gene expression in adipose tissue. We're trying to measure levels in plasma, which is not easy because you really want to use HPLC. But it's been shown by others that angiotensinogen can be expressed in fat, and especially in the setting of obesity, up to, I think, 50% of circulating angiotensinogen can be fat-derived. And so if, in fact, our model is correct, what we see is that when you delete PRDM16, you have increased angiotensinogen mRNA levels in the fat. Perhaps that leads to then activation of the RAS system, and that drives hypertension. So if that's true, we can test that pharmacologically. And the way we can do that, of course, is with an angiotensin receptor blocker. You can inject these mice with losartan, for example. And if the increase in blood pressure is being driven by activation of the RAS system, we would expect to see the map converge so that the difference would go away. And if it's driven by some other phenomenon, and we're considering a number of things, then we wouldn't expect to see that converge. And so these are studies that we're beginning to do right now. So that's just one example of a candidate factor. Of course, we have a variety of other factors, and we're interested in sorting them out. So as I mentioned, we're going to be looking at these orthogonal data sets, either looking at different kinds of fat cells, which we've already done, overlaying that with what we can detect in the blood when you inject mice with azidohomoalanine, and then overlaying that further with a cell type selective labeling. So that will allow us in principle to say, I'd like to look at proteins that in vitro are visceral fat enriched, present in the circulation, and through this model, present in the circulation and specifically produced by fat cells or by thermogenic fat cells or altered in the setting of obesity. And this just shows our sort of framework for how to categorize these factors. We plan to do functional analysis initially in vitro with gain and loss of function when possible to look at different readouts. But then also eventually, because we're looking at whole animal organismal phenotypes, this will require in vivo analysis where we'll need to either inject animals with adenovirus or adeno-associated virus or generate recombinant protein or genetic models to study the effects in animals. And so ultimately, the goal, of course, is to better categorize the secretome of individual fat cells. And so over the long term, if we're successful in this approach, we hope that we will be able to break the link between obesity and disease. And these are a great variety of diseases. I showed you data with hypertension. We're also doing studies looking at diabetes, fatty liver disease, and breast cancer. And so the long-term goal, of course, would be not so much to target obesity or body weight per se, but to break the connection between obesity and these associated diseases. And so just in closing here, I want to thank the people in my lab who did this work. The work on the secreted proteins has mainly been done by Chan-Hee Choi, an MD PhD student, along with Audrey Crane. The work on blood pressure has been done by Sarah Marks, who's a medical student spending a year in the lab. And then we've really benefited from a variety of excellent collaborators. So the work on PRDM16 was done when I was a postdoc in Bruce Spiegelman's lab, and we collaborated with Jerry Schulman at Yale. The work on hypertension has been with the close involvement of Alan Mark, who's affiliated with the University of Iowa, but has also been a guest investigator at Rockefeller for a number of years, along with Robin Davison and Scott Butler at Cornell. And then the chemical biology that I talked to you about has been partly in collaboration with Howard Pang, who's a colleague at Rockefeller. And so with that, I thank you for your attention, and I'm happy to take any questions.